Okay, so we had a pure breeding. Oh, look, you're going to get to see my artwork.
Mm-hmm.
We had a pure breeding plant that makes leaves in its flowers. And so you wouldn't initially know which allele is dominant and which allele is recessive. So instead of using capital and lowercase, because that's cheating, because we all know what those mean, I'm just going to say that that plant has allele L1. So if it's a pure breeding plant, that means if you self it, it only has offspring that look like it, which defines that the genotype is homozygous. Because if this was a heterozygous plant, if you selfed it, you'd get a variety of offspring.
Yeah, I understand that.
1 to 2 to 1 ratio, Punnett square. Okay. So if you cross that individual to an individual that's pure breeding for flowers that do not have leaves, that defines that individual as being homozygous. So we'll just say it's homozygous for a different allele 2. And the only reason that using pure breeding parents is critical is because that's the only way we know what the genotype of its offspring is.
So if it's not purebred, then you wouldn't really know?
Right.
Okay.
So for example, remind me, I'll go back into an example. But yes, if it's not pure breeding, then we don't know its genotype.
So you'd have to assume that there's both in the mixture.
Right. You'd have to assume there's some allele 1s and some allele 2s in an individual. So, okay, just briefly. So I address your question now. We know now that the leaf in the middle of the flower is dominant.
Mm-hmm.
We talked about it in class. We'll get there in just a second.
Yep.
But knowing that it's dominant, what's the other possible genotype of this plant on the left? There are 2 different genotypes that could make it leafy-flowered. If the L1 allele is dominant and produces leafy flowers, what's the other genotype that plant could have?
Little L.
Heterozygous. In this case, L1 over L2, right? Because we've said that L1 is dominant.
Mm-hmm.
So either of those 2 genotypes would produce, either this genotype or that genotype would produce a plant that looks like that. Because there's 1 dominant allele, 2 dominant alleles, doesn't matter. If you've got at least 1 L1 version of the gene, that's causing leaves to grow out of your flowers. The reason that we have to have pure breeding plants then is because that lets us figure out what the genotype of that plant is. If it's pure breeding, if it only produces offspring that are exactly like itself, then it can't be a heterozygote. Because a heterozygote, if you self it, would produce a quarter of its offspring. We could work out the Punnett square, would be L2 over L2. Then they'd look like the plant on the right. So that's the only reason that pure breeding is important, is because we know for sure that the genotypes of the parents are homozygous, which means we know for sure that their offspring is a heterozygote. Because they can only get L1 from the parent on the left, they can only get L2 from the parent on the right. So then the big question mark is, what's the phenotype? You've got 1 of each allele, so whichever allele is dominant is going to show you, if L2 is dominant, it's going to look like a no leafy-flowered plant. If allele L1 is dominant, then it's going to look like a leafy-flowered plant.
Mm-hmm.
And what we observed was, indeed, there were leaves growing out of the middles of the flowers, and so that defined L1 as the dominant allele. L1, I don't know if that's actually common genetic nomenclature, but I just use the greater sign. L1 is dominant to L2. And in that case, then we could translate that, if you want, for future work, to the dominant allele is capital L, and the recessive allele is lowercase l. But that's the first time now that we can actually say capital L, lowercase l. We've experimentally determined which allele is dominant. Is it the parent on the left or the parent on the right? It's whichever allele the parent on the left had. So that gets us to the most important information, which is that the F1s, so here's the P0 generation, F1 generation. So the F1s are all heterozygous.
Okay.
And we only know that because the parents are pure breeding. If you cross two pure breeding individuals, you make a heterozygote. So what's the next part of the question? So we figured that was question 1, which phenotype is dominant?
Use the F1.
So self the F1. Let's start over. So we've got an F1, it's capital L over lowercase l. And that is, the phenotype is leafy-flowered. So when you self that individual, what are its offspring's genotypes and phenotypes? We don't really need the genotypes, but we're going to get the genotypes before we figure out the phenotypes. That is, we'll know what their genes are before we learn what they look like. We have to know their genes to know what they're going to look like.
Since you have a mixture of both, wouldn't it be 50-50 on both sides, right?
Exactly. So you've got one plant, or one parent. So whatever is written on the top is going to be written along the side. This is the same individual. And you've got, 50% capital L gametes and 50% lowercase l gametes. So fill that in by copying across and copying down. And we get genotype ratio and we can get phenotype ratio. And I always start with the genotype ratio. It's usually more cumbersome, but there's one of those. That's the upper left square. There's 2 capital L over little l. The upper right corner and the lower left corner. And then there's 1 lowercase l over lowercase l in the bottom right. So we've got a 1 to 2 to 1. For every 1 of those, there's 2 of those, and then there's 1 of the recessive.
That's the genotype.
This is the genotype ratio. So now what we have to do is calculate, or convert that really, into a phenotype ratio. What do these plants look like? Or what do we predict that they look like? So how do we go about doing that?
So, do you want the actual description or mathematics?
Oh, the description. Phenotypes. Yes. What are the 2 phenotypes?
Well, the 2 phenotypes is you have the possibility that it's a plant that has a leaf growing out of the middle of
Yep.
it and displays white petals, correct?
Yep.
And then the other is you have just the petals, no leaf, and so it just looks like a basic flower.
Right. So it's the 2 parents, basically.
Yes.
It's
Or the grandparents.
There are 2 grandparents. You're right. There are 2 phenotypes, either leafy flowers or non-leafy flowers. So what genotype gets you leafy flowers?
Yeah, this one. But this one as well.
Right. And we only know that because we determined that because we crossed the pure breeding parents and found out that capital L is dominant.
Yes.
So any of these genotypes that has at least 1 capital L is going to produce leafy flowers. And that's the sum of that 1 and 2.
Okay.
So that gives us 3. And then you get 1 individual that's wild type. So you get a 1 to 2 to 1 genotype ratio. But when there's dominance, you sum together that 1 and the 2 to get 3, and that's how you get the 3 to 1 ratio, phenotypes. So that's 1 to 2 to 1 genotype ratio, 3 to 1 phenotype ratio. And that's what's common for a monohybrid cross. You cross 2 individuals or self 1 individual that's heterozygous.
So if it's not a monohybrid cross, how would it differ? Out of curiosity.
Let's do that after we finish. Is that okay?
Yeah.
Okay. Remind me if I forget.
Okay.
Which I shouldn't, but might. I might get distracted by cool geneticsy things. So, to answer the question now, I'm going to switch to red for the rest of this.
Video is 456.
Okay, so we've got self the F1s. The total of these that 3 ratio, leafy-flowered plants, we produce 456. We've got 456 F2s. All right, I should label that. That's the F2 generation. 456 F2s that have leafy flowers. And the question is, how many plants do you expect to see that don't have leafy flowers?
Okay, so that makes sense why you have to do it divided by 3, because you're trying to eliminate this out of the equation.
Right. So this 456 is 3/4 of the area of the Punnett square, and 1/3 of that number, or 1/3 of that area, gives you 1 unit of area.
Yeah.
This is the way I think about it. This is 3 parts, so if you divide that by 3, that tells you what 1 part or 1 unit is. And that is whatever it is we said in class, 152. So it's a 456 to 152 expected ratio, which if you divide by 152 each side, you get back to the 3-to-1 ratio.
That makes sense now.
I just
Okay.
wanted to check my
Yeah.
on that, because I was doing it
Fantastic.
in class and I was like, wait, I don't think it looks right.
So if you had a leafy flowered parent P0 cross to that, and this was, capital L over lowercase l, cross 2, we know that has to be homozygous recessive. Okay. So if we hadn't started with a pure breeding plant in the P0 generation, how badly would that screw us up? Is basically the question.
Yes.
So, what are the offspring?
So you've got a lot more little elves. The offspring might demonstrate the recessive gene since it's so dominant, technically.
Yeah, so what's going to happen is one of these parents, you don't know which, of course. We know because I wrote out some letters, but initially, doing this experiment, you wouldn't know. One of the parents only produces little L gametes. That's that parent.
Yeah.
And the other parent then produces 50% capital L and 50% lowercase l gametes. So then you get a 50-50 mixture of leafy flowered plants there and 50% non-leafy flowered plants there. And then basically you've got plants of both types, so that doesn't tell you anything about dominance. It's like you started with 50%, one of your 2 parents was leafy and one was not. And then you wound up with 50-50 kids that have one phenotype or the other. That's why the pure breeding part is important. If you only knew, but that's why we do it, if you knew which were the individuals that were heterozygotes, then you would know that it's their phenotype that is the dominant one. But because then for every one of those, you generate one of those, you can't empirically distinguish, well, which one is the phenotype I'm supposed to be looking at. I've got both phenotypes in the F1 generation.
Yeah, that's why I kind of asked the question.
Yeah, yep. So that's what happens.
So if you can't differentiate because they're on equal plane when it comes to ratio, how would you structure that out if you were actually asking for calculations?
I wouldn't on this question.
Okay. Okay. I was
Oh my goodness.
I was so worried when I was looking at that.
I mean, I could formulate a question about this, but I would give you all of the genotype information.
Okay.
So the point of this question that I asked in class, that we went over in class, was the points were can you do phenotype and genotype ratios? Can you set up Punnett squares? And maybe most importantly, can you convert from the wording of the question? So when I say something like pure breeding, do you know that means homozygous?
Yes.
So it's all about nomenclature, and can you convert a word
Into an actual thought,
Yeah,
into an actual action.
Can you convert a word question or a written question into genetic nomenclature?
Got it.
So if you're remaining heterozygous, how would you word it instead of saying pure breeding?
You wouldn't put.
I would just say I would say non-pure breeding. Yeah, I might just say heterozygous.
Okay.
Check the mock exam. I think it's not a question I wrote. It's one of the questions that one of the students submitted was a really well worded, I thought, genetics question, and I think it maybe gets to that. In fact, I might as well not be so obtuse about it. I can just pull up the mock exam and show it. Oops. Except that's the first exam. So question 6, kind of more practice on that word questions, converting written descriptions into genotypes. So that would be a good one to look at. To practice these sorts of approaches. And also, question 10. Where, it did specifically say you cross homozygous recessive fly to a heterozygote. It's hard to say heterozygote in any way other than just saying heterozygote. Like I said, the only other way I could think to state that would be to call it a non-pure breeding genotype, which is kind of clumsy. I might as well just say heterozygote.
Yeah.
But the way I could phrase that if I wanted to write a long written question would be, you self an organism and it produces 2 phenotypes of offspring. Then that would be your clue that that parent wasn't pure breeding. Therefore, you would say, oh, that's a heterozygote. That would be the really long, tortuous way to put it.
So just read the question carefully.
Yeah, basically. And ask questions if you have questions with. There are some questions I won't answer about interpreting exam questions. Sometimes, if you're asking a question about what I'm trying to test you on, then I'm not going to help you interpret the question, but I'll just say I'm not going to tell you. So it doesn't hurt to ask, in other words. Sometimes it's helpful. At worst, it's neutral, and it kills a half a minute of time, which could be bad. But yeah.
I think for this one, I was just thinking too much into it, like the whole process, because I actually work with the plant breeder. Like, that's my job
Cool.
when I graduate.
Nice.
So I was like, oh, well, I don't know, what if you're getting all the seed, and how do you get
Oh my goodness.
So I was like, no, I'm going way too into it.
Hard to separate professional life from class life.
Yeah, so I was like, I don't understand this, and I should understand this.
Yeah. No worries.
Yeah, so I think I was just thinking too much into it.
So when we're being tested and we're expressing genotype and phenotype ratio, if you do ask us additional questions, it'll be something simple like what you gave us at the end, correct?
At the end of this question.
Yes, where you have like 456, right? 6 different variations, and then you just have to try and plug in the ratio into the ending. Okay.
That's a good example question for the sorts of things that could be asked. And I can't ask you anything that's impossible to do. Well, I could, I suppose, but that would just be... I'm not that mean. I like to think I'm not mean at all, but I probably am in some ways. I do write purposefully difficult questions, but...
Well,
I try not to be unfair, let's put it that way.
Yeah.
Try.
Could I ask you about number 5?
Sure. Mock exam number 5?
Yes.
So the dihybrid parent from the mock exam, question number 5. We'll produce all 4 different combinations of capital and lowercase e's and capital and lowercase f's. So capital E with a big F, capital E with a little f, lowercase e with a big F, and lowercase e with a little f. And then you just fill down and fill across. And then the only thing you have to do is check to see are there the same genotype in multiple squares in the Punnett square
Okay.
to get the ratio.
So you are just asking for Punnett square and ratio in that question?
Yes.
Okay.
And just genotype ratio.
Just checking.
'Cause I haven't had any phenotype
Oh,
information, so I
sorry.
can't tell you anything about, or I can't ask you about phenotypes.
Okay. And did you want general description in words or just the phenotype?
Just so, how many different genotypes? If I was answering this question the way I wrote it, I would have answer A is how many different genotypes are there? I think there are 6 in this Punnett square, which has 8 cells, but I think there are 2 duplicates. So I think there are 6 different, if I remember correctly, I think there are 6 different genotypes. That would be the answer to the first question. And then what are those genotypes and what ratio are they in would be the answer to the second question. So that would be writing out all the genotypes and showing what the ratio is between them. So that would look something like, this is not true for this question, that would look something like 1 to 2 to 1, capital E over capital E over lowercase e, and lowercase e over lowercase e. That's how I expect genotype ratios to be written, for example. So the ratio and then the genotypes, or if it's a phenotype ratio, the numerical ratio, and underneath or above it, what are the phenotypes that are associated?
Okay.
So the semicolon means different chromosome, right?
Yes.
Semicolon.
So these semicolons mean that those are on different chromosomes. Yep. And the only reason that's important and the only reason the slashes are important to tell us that these are diploids, the only reason that the semicolons are important is because at some point we start talking about genes that are on the same chromosome, and then we get rid of the semicolon. And that's coming up soon. So it seems extraneous now because we've only been dealing with cases where genes are on different chromosomes. And it turns out that's only because that's the situation in which Mendel's laws work the best. So Punnett squares and all of that work fine as long as genes are on separate chromosomes. Once you start putting genes on the same chromosome, then all hell breaks loose. So I'm just doing this because the semicolons, because I'm explicit. I want to make sure everybody knows that we're still talking about genes on separate chromosomes. That way when we start talking about linked genes, it's clear that there's a difference. I start leaving the semicolon out. That's why I get so adamant about writing the notation with the semicolon up until the point when we start talking about linkage, because otherwise everything gets really confused. Not that it isn't already, but more confused.
Because it would just be, or you not ask for that.
So for question 4 on the mock exam, that's a great question. What am I asking for? So I'm asking about gamete haplotypes. So how do you figure those? What am I asking? Yeah, good question. So you've got an individual that's T over T. What do they use? Capital Y's. I pick letters where their upper and lowercase forms are a little bit easy to tell apart. No O's. If anybody ever uses an O as a genotype, they're crazy. You can't tell a capital O from a lowercase O to save your life. Okay, or W's or V's or X's. It's all A's, B's, no C's. C's are, anyway, sorry, I'm getting distracted.
You would have liked my biology introduction teacher then. They used O's for everything.
Oh dear goodness. No. Yeah, why? Why would you do that?
I don't know, we said that every exam. Why? Why?
Maybe it's so that you can argue that you got it right. You say, no, that's clearly an uppercase O and a lowercase O. Well,
Oh, no.
they look the same size. Well, if you get the ruler out, you can see they're slightly different sizes.
We tried, but it didn't work.
So, assume for a second that you're selfing this individual.
Okay.
and you want to set up a Punnett square. So what do we write on the top and along the left side?
The sperm and the
Yeah, the haplotypes of the gametes.
Yeah.
The DNA that the sperm and the eggs contain. That's what this question's asking. What gamete haplotypes are produced? Which is, I'm just translating for you now, the same thing as what you write there
Okay.
and there on the Punnett square. And we did this in class a few times, but if you take a genotype, if you take a cell that has that genotype and you go through meiosis, what is in the gametes that are produced? So we could do that if you want. I'm happy to work through it again.
Yes,
but
please.
that's basically how we get to what everybody may be comfortable with doing straight away, which is making all the 4 possible combinations of T's and Y's.
Oh, okay.
So we 4 haplotypes at the end.
Right. Well, yes. What gamete haplotypes are produced in a dihybrid? Right. So this is dihybrid. So we get capital T. Oh dear goodness, that's a capital Y. And a capital T with a lowercase y, and a lowercase t with a capital Y, and a lowercase t with a lowercase y. And that's also the same, since it's the same individual in their gametes, it's the same across the top.
Okay.
So, meiosis. So if we have a diploid cell that's... Oh, I already screwed up, didn't I? T over T, capital Y over lowercase y. You do that right? Excellent. So there's a diploid cell. We're talking about 2 different chromosomes. Then what's the first step of meiosis? We're going to make some gametes. Start with a diploid cell. Yeah, to do DNA synthesis. S phase in the cell cycle. So every one of those 4 pieces of DNA there. Capital T chromosome, a lowercase t chromosome, and then a separate chromosome pair, we've got a capital Y chromosome and a lowercase y. So after synthesis, every one of those 4 molecules of DNA has replicated. So now we have 8. 2 capital T's, 2 lowercase t's, and so forth. And then glossing over some details, the first cell division metaphase before the first division. Those chromosomes all line up at the middle of the cell. Because what's going to happen is during the first division, if the first division happens down the cell like that, half of the chromosomes go one direction and the other half go the other direction. And sister chromatids stick together. So the copy of the original chromosome and its copy stay together. So the original capital T and the one that was just synthesized stay together at the first division. And go back and look at the videos where I talk about meiosis, but there are 2 ways to arrange these 2 sets of chromosomes. The other way would be to have the capital Y's and the lowercase y's flipped. So this combination is going to produce after the first division then. So here's the first division, division 1. You're going to get the little t's with the little y's in the cell on the right. And you're going to get the capital T's with the capital Y's. Who knows? Anyway. So that's how you get, after the second division, each of those 2 cells divides. So you get your 4 gametes, and now we know for sure you've got little t's with little y's, 2 of those. So each of those gametes gets a little t and a little y and a little t and a little y. And then the cell on the left, you get a big T with a big Y and a big T with a big Y. And if you had, so that's just the product, those are the 4 cells that are product of one meiosis. But for every one cell that went through meiosis that had that arrangement of the 2 chromosomes we're looking at, there were an equal number of cells that had the capital Y's lined up on the side with the lowercase t's. Because this is all chance. It's just how the chromosomes are floating around inside the nucleus. Half of the time they're going to line up like this. The other half of the time, the other half of cells that are going through meiosis, they're going to be just arbitrarily aligned like that. So for every meiosis that looks like that, you get one with the opposite orientation. And if in that orientation, then you get the capital T's with the lowercase y's and the lowercase t's with the capital Y's. Then you get the second division. And then you get capital T, lowercase y, capital T, lowercase y, lowercase t, capital Y. And then notice that these different genotypes exist in a 1 to 1 to 1 to 1 ratio.
How are you doing?
Good afternoon.
Say hi.
Hey, good to see you. Welcome back. So you get, even though there's 2 of each of these cells, it's a equal ratio. For every one of those, there's one of those, there's one of those, there's one of those. And that's why when we set up the Punnett square, there's an equal number of those 4 different haplotypes across and down the side of the Punnett square.
So on the test, can we do only that one, or we have to do both?
Yeah.
Let's see. So this question,
Oh, okay.
so for the mock exam question 4, you're just asked what are the gamete haplotypes, and those are the 4.
Okay.
Yeah, so you would just write that. To answer this question, if this was on the exam, you would just write out, these are the haplotypes.
So
Capital T with little y, with a semicolon in between, by the way. You have to remember to put the semicolons in there. Because that tells us, maybe, or not, whatever. If the stylus won't work, then I can't do anything about it. There. Capital T, semicolon, lowercase y, and so forth.
Do you want us to draw that whole process out?
No.
okay, that's what
No,
my next
please.
question
Yeah.
was.
No. If you're comfortable going straight from genotype of the parent dihybrid straight to the genotypes, haplotypes of the gametes, that's fine.
Okay. So when we're writing it out, do you want it?
I can have some paper.
Yes.
Here.
Oh, thank you.
Sure.